Okay, good morning friends. This is Dr. Doop Singh, lecturer in Botany, Government Degree College, Sitapur Mandi. Today I am going to discuss about the photophosphorylation. So photophosphorylation in the presence of the light, in chloroplast pigment, the adenine dinucleotide phosphate, it can absorb one inorganic phosphate and it modified or it can form as a adenine trinucleotide phosphate. So it is called as a photophosphorylation. So photo mean light. So in the presence of the light, the phosphate is formed. The adenine dinucleotide phosphate is converted as a adenine trinucleotide phosphate. So here one inorganic phosphate is adding to the adenine dinucleotide phosphate. So this type of phosphorylation is occurs in the chloroplast in the presence of the light or photo. Hence it is called as a photophosphorylation. So this photophosphorylation, it is initially expressed by Ammon. Who is that? Ammon. Ammon was he expressed this photophosphorylation. So this photophosphorylation is two types. What are they? So if you can see in the presence of the light, so adenine ADP, adenine dinucleotide phosphate plus inorganic phosphate. So IP mean inorganic phosphate. So in the presence of light and chloroplast. So these reactions occurs in the chloroplast matrix. So chloroplast. So the adenine dinucleotide phosphate, it absorbs this inorganic phosphate and it will be converted as a adenine trinucleotide phosphate. Adenine ATP, adenine trinucleotide phosphate. So this is occurs in only chloroplast matrix. So stroma, not matrix, a chloroplast stroma. So it is called as a photophosphorylation. So this photophosphorylation is first expressed by the photophosphorylation is expressed by Ammon. So here it is of this photophosphorylation is two types. What are they? One is cyclic photophosphorylation. And second one is non-cyclic photophosphorylation. Today and now I am going to explain about the cyclic photophosphorylation. So here cyclic photophosphorylation, I am going to draw a figure about the cyclic photophosphorylation. So here in this cyclic photophosphorylation, actually initially in this cyclic photophosphorylation, the only photosystem 1 it can involve in this procedure, and in this for PS1 or cyclic photophosphorylation, the presence of the light, the electrons of photosystem 1 or photosystem here or PS1 in the chloroplast, it passes through a chain of electron. It carries and loses some energy and return to the same pigment system. So this is known as a cyclic photophosphorylation. So in the presence of the light, the electrons of photosystem 1 in the chloroplast passes through a chain of electron. It carries and loses some energy. And it return to the same pigment system. So it is called as a cyclic photophosphorylation. So this cyclic photophosphorylation, it is first demonstrated by Frankel Frankel in the bacterium Rhodospirillum rubrum. Rhodospirillum rubrum. So it is demonstrated by Frankel. So the cyclic photophosphorylation, it is demonstrated by Frankel in the bacteria. What is the bacteria? Bacteria Rhodospirillum rubrum. Okay. So in the Rhodospirillum rubrum bacteria, so it was demonstrated or expressed by Frankel. So this is expressed by the Frankel in Rhodospirillum rubrum bacteria. Now I am going to draw a cyclic photophosphorylation pathway. So actually in this photophosphorylation, the PS1, it means photosystem 1. So in this photosystem 1, it has the reaction center as P700. So P700 is the chlorophyll pigment A. It is a chlorophyll pigment A. It is it can act as a reaction center in photosystem 1 because the chlorophyll pigment A, which is act as a reaction center in PS1, it can able to receive the 700 nanometer wavelengths of the spectrum. Okay, hence it is. So it would like this chlorophyll pigment of the PS1. It would like to receive. It can receive the 700 wavelengths of the spectrum. Okay, 700 nanometer wavelengths of the spectrum. It can receive. And it can excite and it can release the electron from inner one to the outside. So this is something about the PS1. So here actually, so these are the wavelengths. These are the red wavelengths. So the photon is, so this is the photons, a small minute parts of the radiation are called as a photon. So when a photon reaches to the PS1, so that in the PS1 the reaction center which is called as a P700. So the P700 is the modified chlorophyll pigment. It would like to receive the high wavelength of the red light. So that is the P700 or 700 nanometer wavelength of the spectrum. It can receive and it excited the chlorophyll pigment. It can excite it and it can release one electron from inner radiant to the outer one. So this is the P700 reaction center of the PS1. And next one, this for example, it is the electron, and it comes outside this electron. Okay, this electron it reaches to the FES protein. FES, ferrous and sulfur protein. FES protein. So it would like to, this FES protein, it receives the electron. And it transports to the ferridoxin. The electron it transports to the ferridoxin. And also the ferridoxin it receives two electrons. Ferridoxin it is received two electrons, and two electrons again they transported to the home. So these electrons are transported to the cytochrome B6. and this cytochrome B6 it can transfer the electron to the which, so it can a quinone cycle. It is a quinone cycle. So in this quinone cycle, so it is a quinone, semiquinone and semiquinal, quinal, ubiquinone, semiquinone and ubiquinol. For example, in the quinone cycle, ubiquinol, ub semiquinol, ub semiquinol, and it modified as a ubiquinol. And this ubiquinol it releases electron and it releases proton, and it is again modified as a ubiquinol. So again this is something about the quinone cycle. And next this electron it transfer to the cytochrome C, cytochrome F, not cytochrome C, cytochrome F. Just a minute. Cytochrome F. So cytochrome F2, this electron again it is transferred to the plastocyanin, PC, plastocyanin. So this electron transferred to the PS1, P700. Okay. So here electrons are not utilizing. So these electrons are rotating cyclically. So it is they are rotating in this cycle. So there is no use. So the ferredoxin actually here the ferredoxin should receive the electron here, NAD, nicotine amide dinucleotide. It is the oxidized nicotine amide dinucleotide. It should receive the electron. But when the NADs and FADs are absent in the chloroplastoma, okay, when the NAD and FADs are absent in the chloroplastoma, at that time these ferredoxin that cannot, the electrons are not utilized. So these electrons they are transported to the cytochrome B6 pump. So and the cytochrome B6 that can transfer the electron to the quinone cycle. So in this quinone cycle, the hydrogen water is not oxidized here, water is not photolysed. So this presence of the light electron PS1 in the chloroplast. So here it passes through a chain of electron, carries and loses some energy and returns the same pigment system. So okay, next one, when the PS1 is activated, here the cyclic photophosphorylation. In the cyclic photophosphorylation, it involves only pigment system 1 only. It involves only pigment photosystem 1 or pigment system 1 only. So here as there is no photolysis of the water and releasing of the oxygen is also absent during the photophosphorylation. Okay, now it cannot oxygen cannot release in this cyclic photophosphorylation. And next one is what is that? Okay, the photolysis of the water. Photolysis of the water is cannot happen. So due to these two processes, so here only what will happen? Here only the ATPs are formed in this procedure. The photolysis, so photolysis of the water is not continue, and also the oxygen is also not released in this procedure. So when PS1 is activated by one photon, PS1 is activated by one photon, the reaction center P700. What is the reaction center of PS1? So P700 is nothing but a chlorophyll A pigment. Just now I told you. Okay. So this reaction center of P700 is excited. It is excited and it releases two electrons. So this electron is first carried by ferrous sulfur protein. This is the ferrous sulfur protein. So these are the first electron receiver in the cyclic photophosphorylation. So the ferrous sulfur protein is the first electron receiver in the PS1, and also the ferrous sulfur protein, it reaches the ferridoxin. It reaches the ferridoxin of oxidized state. So as a result, the ferridoxin in reduced. Ferridoxin is reduced. So ferridoxin is reduced. So as a result, it is reduced, and then the electron returns from reduced ferridoxin to plastoquinone. The electrons, they are reduced. Actually, these electrons should utilized by oxidized NAD and FAD. So when the NAD and FADs are zero in the chloroplast stroma, at that time these electrons are transported to the cytochrome complex. So in the cytochrome complex, that electrons are transported to the cytochrome B6 complex. So here this electron, it returns from the reduced ferridoxin to plastoquinone. So these electrons from the cytochrome B6, it is transferred to the plastoquinone. So the plastoquinone through the cytochrome B6 from the cytochrome B6, the plastoquinone, it receives the electron and it reduces to the plastoquinone, semi-plastoquinone, and semi-plastoquinone again it receives two protons, and that can reduce as a plastoquinone. So the protons of the plastoquinone, the protons are translocated through the plastoquinone pole. So these are released into the lumen of the thylakoid membrane. So these plastoquinones, when the plastoquinone is again oxidized, so the two electrons and two protons are released from the plastoquinone. So these protons, they are released into the lumen of the thylakoid membrane, and now the electrons are transferred to the cytochrome F. The electrons are transferred to the cytochrome F, and from there it reaches the plastocyanin. So these electrons again they transfer to the plastocyanin. So plastocyanin and plastoquinone is the motile electron carriers. These are the mobile electron carriers. So they would like to move from one place to another place on the thylakoid membrane. So this plastocyanin finally it receives the electrons, and this plastocyanin it receives the electron, and it directly the received electrons it is transferred to the P700 of the PS1. So P700 of the PS1. So one ATP is synthesized in this electron transfer from the ferridoxin to the cytochrome B6. So one ATP, when electron with the one ferridoxin to cytochrome B6. So here here when the ferridoxin is there, so here to here when the cytochrome B6, when the electrons are transferred to the ferridoxin to cytochrome 1, so one ATP is formed in this process. So here this type of phosphorylation is called as a photophosphorylation. And ATP is synthesized in the electron transfer. ATP is synthesized in the electron transfer from plastoquinone to cytochrome. One ATP is synthesized in the electron transfer from the ferridoxin to the cytochrome B6, and one ATP is synthesized in the electron transfer from the plastoquinone to the cytochrome F. So this is something about the cyclic photophosphorylation. And next one is in cyclic photophosphorylation, two ATPs are found. In cyclic photophosphorylation, there are two ATPs. When the ferridoxin, the electron transferred from the ferridoxin to the cytochrome B6, here one ATP is formed, and another one is the electron when transferred from the quinone to cytochrome F, here also one ATP is formed. So totally in cyclic photophosphorylation there are two ATPs are forming. But in this procedure, in this non-cyclic phosphorylation, in this cyclic photophosphorylation, the ATP, the releasing of the oxygen, releasing of oxygen is absent. So oxygen it cannot release because the photolysis of the water is not happening in this procedure, because electrons are not utilizing. So electrons are not utilizing, so there is no deficiency of the electron, because the released electrons, they are surrounding, they are transferred from one electron carrier to another electron carrier on the thylakoid membrane. So these electrons are not utilized, so they are transferred in a cyclic manner. Hence there is no deficiency, there is no occurring the deficiency of the electrons. Hence it is called as a cyclic photophosphorylation. So in this procedure two ATPs are forming, but photolysis of the water is not occurring, and also the oxygen is not releasing. When the photolysis of the water takes place, automatically, what is that? For example, here, 1 minute. If we here water H2O, two molecules of the H2O in the presence of the radiation, sunlight, so it is converted into 4 protons plus 4 electrons plus 1 oxygen, 1 O2. So this O2 is released by the photolysis of the water. So it is not happening in the cyclic photophosphorylation. So this is something about the cyclic photophosphorylation, and tomorrow I will discuss about the non-cyclic photophosphorylation. Thank you very much for this class, who are attending the class. Thank you very much.
Please